I'm Daniel Levine, and this is the Bio Report. Drugmakers have employed new technologies to reduce the cost and time it takes to develop drugs and produce therapies that more precisely target the underlying biology driving diseases. Boston-based Berg is using artificial intelligence with a variety of other technologies to re-engineer the discovery and development process. We spoke to Niven Narain, co-founder, president, and chief technology officer of Berg, about the problems with drug development today, what makes Berg's approach unique, and what its pipeline can tell us about whether the approach is working. Niven, thanks for joining us.
Well, thank you, Dan. It's great to be with you.
Let's start with drug development today. What would you say is wrong with the way most companies go about developing new drugs?
I would say that most companies make, obviously, a fervent effort to create drugs in the best way towards helping as many patients as they can. But I think one fundamental issue within drug development, as it is today, is that we still are, as an industry, we're putting a chemical screening up front instead of putting the biology up front. We don't often learn about the effect of these chemicals on a patient's biology until the drugs have hit the market at a significant level. And of course, most drugs that are in development, I think it's 1 in 10,000 molecules that are screened actually go into clinical development, and 1 in 100 phase 1s actually become an approved drug.
Your company, Berg, has built a drug development platform. What's different about the way Berg is approaching the problem of drug development?
So Berg has made a conscious effort based on the tenets that I just mentioned to actually go back to biology. So what we do is we make no assumptions up front. We actually somewhat are flipping the scientific method upside down, where we are allowing the data to generate the hypotheses instead of the hypotheses generating data. So we start all of our processes up front by screening human tissue samples from individuals with a given disease, for example, cancer, and then we take, if at all possible, from that same patient, normal healthy tissue or a separate cohort of healthy individuals, where we then drive a process well beyond genomics by looking at that patient's genes, the patient's health records, and other types of omics that we refer to internally as a phenome or phenotypic medicine, to look at their proteins, metabolites, and really to understand as broad as possible the patient's biology and narrative. So then the decisions that are made to focus in on what has gone wrong can be appreciated and illustrated by the use of the artificial intelligence program that we have, where all the data is fed into that AI, and then at the end of that process, what comes out looks like a map, and that map on the patient populations allows us to understand which genes and proteins are governing the difference between health and disease.
So you made a reference to some of the technologies you're employing. There are a number of companies that have used various omics. They've used in silico drug development. They've used systems biology approach. What's different about what Berg's doing from what's been done elsewhere and in some of these next generation drug development firms?
Sure, no, that's a fair point. And then the difference, as compared to some of the other companies that are focused very deeply and with significant expertise in individual verticals or individual silos, or companies, as you mentioned, are proteomics companies or bioinformatic companies. Berg really puts the tissue sample at the foundation, and then what happens is that we screen the genomics, the proteomics, metabolomics, the lipids, and that information is then intercalated in an agnostic way and subjected to this Bayesian AI system that then allows all of these processes to occur within that one sample, so that we're integrating all of these data sets in a manner that helps us to understand much more fundamentally about that sample. But the second layer is that expression or an understanding of expression of genes or proteins, because of the nature of biology, does not always mean that it's going to translate into a loss of function. So we add on a second layer by looking at things in the biology that pertain to mitochondrial function or energy production, inflammation, etc., where we then perturb these tissues or cell models in a manner that allows us to appreciate what happens in this mimicked environment of disease. So we're capturing expression, we're capturing the functionality, and in most cases we have the clinical outcome data. So we're spanning these patient populations all the way from the cellular basis, the functional model, and then of course in the outcome that we have from the health records.
So it seems like these new approaches provide great opportunities for more intelligent and elegant drug design and drug development, but we're still quite limited in our understanding of the variosomics of gene networks and the networks of disease pathways. Does your approach somehow get around the limits of our understanding today?
I think, Dan, it helps to unravel new understanding. I think that most of, there was a Nature paper that came out in the past year that showed that many of the targets that are published, or many of the drugs that we have now even marketed, do not actually hit the original target that one had suspected. So we, as I said, by taking the agnostic approach, we're able to learn more about the biology. But biology in and of itself is fairly messy. Even in a healthy environment, it's fairly messy. And I think that in an effort to strategically clean up the biology, we actually lose some of the real fundamental underpinnings of what is leading to disease. So by the Berg approach of encapsulating and including all of the data sets with no cutoffs, etc., we're then able to allow this noise, if you will, within the biology, the messiness of the biology, to be captured by this supercompute AI platform that one could not obviously humanly appreciate, but the AI helps us to make sense of all of this data.
Before, you were talking about the way traditional drug development's done, where a chemical library will be thrown at a target. You don't use chemical libraries or synthetic compounds. Instead, you focus on organic compounds that are naturally occurring in the body with the intent of normalizing the disease microenvironment by essentially using the body's own machinery to fix itself. Can you explain this and why it may be a more effective or beneficial approach?
Sure. So, and that's absolutely right. So Berg has not employed the use of chemical libraries. The approach is that since we fundamentally are starting with the human tissue sample up front, instead of learning what's going to happen to the patient in the clinical trial or post-market surveillance, we're moving that piece of the puzzle to the forefront, and by creating the differential between what's with the healthy model that's built and the disease model, the difference is really the things that have gone wrong, which genes and proteins and metabolites, or etc., are responsible for the onset or the proliferation of a given disease. So what we do then, after these models or these maps are built, if you will, like take the American Airlines map, for example. Americans, since the merger with U.S. Airways, they're now headquartered in Dallas with hubs in Phoenix, and Kennedy in Miami. So we know that for the pressure points that govern this airline are in those 4 cities. So when something goes wrong in New York airspace, you may be flying from Topeka, Kansas to Portland, Oregon. But if your plane is at LaGuardia, something's gonna, it has nothing to do with those 2 cities, but you're gonna end up in a delay or a cancellation. And disease and the networks of biology work as such. So what we're able to appreciate from these models is which of these genes and these proteins metabolites are causing these deviant changes in the biology. So then when we go back and we go back into the wet lab and validate these findings by either doing a knockdown of that gene in a cancer model to see then if you knock a certain gene or protein down, do we actually see that the cancer cell now is undergoing apoptosis, or are we seeing that in skeletal muscle if you're able to redeliver a certain protein or enzymes into skeletal muscle? Are you now seeing that you're increasing insulin sensitivity in a diabetic model, or increasing glucose uptake? So all of the surrogate endpoints of given diseases are tested in the wet lab once these maps are delivered on these patient populations. So then the approach is to use those strategies that are now validated in the lab and put them through now a traditional pharmaceutical clinical development program, whether it's by employing the use of antisense oligonucleotides or siRNA technologies. If we have to do a knockdown, or in the case of an overexpression, we can then redeliver enzymes or peptides, proteins in a manner that's pharmaceutically acceptable, where we can even in some cases target the organ of interest, so that we can then use, and that's where that term, using the body's own machinery to fix itself, because we're essentially just rebalancing or creating that homeostasis of levels of that given biological molecule to treat disease.
So it's still relatively early days for the company, but has there been any proof of principle yet? Is there a case to be made for this approach saving time or reducing the cost of drug discovery and development?
Yeah, no, that's a great question, and we our latest and most mature asset is in oncology, which is BPM 31510, and that molecule, which is a ubidecrenone based technology that has been made into nanoparticle suspension, is meant to drive and change the metabolism of the tumor microenvironment by re-energizing the mitochondria to behave much like a healthy mitochondria. And when you have healthy mitochondria, cell death is a normal part of health. Or skin cells replenish every 28 days, and this molecule is able to switch the fuel that cancer is depending on, and reinitiate apoptosis. So we've now seen, even in our clinical trials, we're seeing proof of principle in 2DG PET scans that when the drug is administered to patients, we're seeing that the metabolism of their tumors are actually being mitigated and decreased. And in addition to some other clinical endpoints and benefits. So in our clinical trials, we're seeing the validation of this mechanism from cell to the preclinical models to now in human trials. And then we also have novel prostate cancer markers that have come out of this platform that are ending a CLIA validation for the launch of a CLIA test. We've tested this in prospective tissue and sera samples in collaboration with the Department of Defense, and thirdly, we have some preclinical diabetes drugs that have been validated in preclinical proof of principle animal models. So those are three of many examples that show proof points and validation points of interrogative biology, but those are the three most mature assets of the pipeline.
And in terms of the lead, which is a topical treatment for skin cancer, is that right?
Yeah, so it's in two dosage forms. We have a topical form indicated for skin cancer, and then we have an intravenous form that is indicated for solid tumors.
So how long did it take you to get where you are today?
We started the first trials in solid tumors in 2011, and we've been able to develop, identify the target, develop the dosage forms in less than 5 years, essentially from the time that the platform was able to help to guide the mechanism of action and development. So we went from discovery to validation, preclinical and clinical in less than 5 years.
Berg takes its name from Carl Berg, a billionaire who made his fortune in real estate. I take it his backing has provided an unusually stable financing that has allowed for the company to build and develop itself differently than it might otherwise had it been backed in a more conventional way. What has this allowed you to do differently?
Yeah, so you're absolutely right. The very unique and supportive funding that's come out of Carl and the other co-founder, Mitch Gray, has just allowed us to scale the platforms that I spoke of earlier in omics and building the Bayesian artificial intelligence, the supercompute capability, in a manner that's allowed us to be much more innovative and to spray outside of a traditional vertical and what you would usually see in an early startup biotech. It's allowed us to span multiple diseases at one time, namely CNS diseases, diabetes, and cancer, and allowed us to really attract significant and unique talent in all the verticals relative to systems biology, artificial intelligence, leading experts in the fields that we're focused on, but just allow innovation to go beyond the usual confines that you would see in an early startup.
So Berg seems to have a rather ambitious development program. It has a relatively broad pipeline that includes not only oncology, but metabolic disease and CNS, as well as diagnostics, as you made reference to. These programs are in cancers and autism and Parkinson's. What's the development strategy? How are you going to prioritize and make sure you have adequate resources to advance your pipeline? And what role do you see partnering playing in terms of your long-term development strategy?
Right. So we really see ourselves as being adept in the early components of the drug development process. We see our business model is to really stay focused on discovering and validating and developing these therapeutics up to proof of principle in phase 1, phase 2, and in diagnostics up to CLIA and potentially further on through partnerships with 510K. But we see ourselves as really an organic feeder into the pharmaceutical industry, where we can then partner with big pharma, with mid-sized pharma to commercialize and continue the late-stage trials all the way to the NDA. We don't have any intentions of creating a sales and marketing teams. We really want to just focus on the front-end innovation and clinical proof of principle. So our business model is through strategic partners that allow us to create the types of co-development deals, the type of deals that allow interrogative biology to span that life cycle from the discovery of a target all the way to post-market surveillance as a technology partner, even after we've licensed the technologies or partnered them out, so that the late-stage trials and NDAs can be filed.
Niven Narain, President and Chief Technology Officer for Berg. Niven, thanks for your time.
Thank you so much, Dan. I appreciate it.
Thanks for listening. The Bio Report is a production of the Levine Media Group. To automatically download this podcast each week, subscribe to our RSS feed or through iTunes or other podcast manager. To join our mailing list, go to levinemediagroup.com. We'd love to hear from you. If you want to drop us a line or are interested in sponsoring this podcast, send an email to danny@levinemediagroup.com. Special thanks to Jonah Levine, who composed our theme music, and the Jonah Levine Collective, which performs it.